Okay, dear colleagues, thanks for being online with us. It's my pleasure to open today's BIH lecture, and it's a great pleasure to announce Hans-Peter Kiem, who has a long, since a very long time, a good colleague and friend in the field of cell and gene therapy. We met like 25 years ago for the first time, and since maintained a very strong interaction. Hans-Peter is, as you've seen, associated with the Fred Hutchinson Cancer Center, where he leads the cell and gene therapy program. And he has, in his career, successfully trained a number of good colleagues in the field, some of which also returned back to Germany and continued their career here. The impressive thing about Hans-Peter is that his research and activities as an MD cover the entire spectrum of the value chain, from ideation over preclinical studies in various very advanced preclinical models, including large animal models, through to initiation and conduct of clinical trials in highly challenging fields of cell and gene therapy. And he's also worth mentioning, successful founder of a successful new company that already has more than 80 employees. So he's really aware of all the things that you have to cover when conducting successful translational research for hopefully the benefit of patients. One thing that is very important to mention is that he's also currently serving as the president of the American Society of Gene and Cell Therapy, and that gives you a good guess how well he's connected and how strong his contributions are, not only in the United States, but also far beyond. So indeed, we have, if we are successful with the current grant application, strong hope that Hans-Peter will be with us in the next 3 years quite frequently, because we applied for a visiting professorship, allowing him to come to Berlin more frequently. And right now he's actually with us, sitting here in the headquarters in the room Domblick to give his lecture online. So Hans-Peter, it's a great pleasure to have you here again. I'd like to open now and give the mic to you to start your talk about targeting hematopoietic stem cells for ex vivo and in vivo gene therapy, which is like the holy grail in the field of hematopoietic gene therapy that you will be talking about.
Wonderful. Thank you, Christopher, for the kind introduction. Just making sure, can everybody hear me?
Yeah, we can hear you well.
I see, okay, wonderful. Yeah, thank you so much again, everybody, for coming and listening today. So you've heard the title, and in my next slide, I just want to give you a brief outline here of what I'd like to cover today in my presentation. Again, I'd like to talk a little bit about the progress in hematopoietic stem cell gene therapy, and then also address some of the current limitations that my team has been working on. Talk about hematopoietic stem cell characterization, engraftment, and targeting, and then gene editing approaches for particular disease applications. And then really also talk a bit more about more scalable and accessible approaches moving into in vivo gene therapy. Now, there are many diseases and conditions that have been or could be treated with hematopoietic stem cell gene therapy, and I will show some examples. But really the largest populations and patients are patients with sickle cell disease and thalassemia, or people living with HIV. And the highest incidence, morbidity and mortality, are in low and middle income countries. So this is also why we've been thinking quite a bit about how to make gene therapy more accessible and potentially be able to move these gene therapy approaches also to these low and middle income settings. Most current hematopoietic stem cell gene therapy trials target hemoglobinopathies. And here's just a list, maybe a little bit outdated, but of the various trials, both for lentiviral vectors and also for gene editing. As you can see, there are 7 lentiviral vector trials and 2 gene editing studies currently ongoing. One with zinc finger, one was with zinc fingers, and the other one with CRISPR-Cas technology. Now here's the progress. Really, this has been quite incredible, I think, again, in the sickle cell disease and thalassemia space. On the left, you'll see the studies by Bluebird Bio using a lentiviral vector expressing a modified beta globin gene. So they have treated now more than 100 patients with transfusion-dependent thalassemia and sickle cell disease with really remarkable efficacy, as you can see there. In patients with transfusion-dependent thalassemia, it led to 90% transfusion independence, and in patients with sickle cell disease, there were no severe veno-occlusive events after gene therapy. So that is really remarkable. Of course, this, as you probably know, led to the BLA Biologics License Application approval for thalassemia with Zynteglo just in August, and the BLA submission for sickle cell disease is planned in early 2023. So really exciting news for the gene therapy space and gene therapy field. Now, on the right, for gene editing, you see the gene editing study with the CRISPR-Cas technology. And there, most studies have actually used fetal hemoglobin reactivation based to some extent, and I'll talk about this later, on the observation made by actually George Stamatianopoulos, the founder of the American Society of Gene and Cell Therapy, that patients with hereditary persistence of fetal hemoglobin had significantly improved outcomes. So here on the right now, you see the study by CRISPR and Vertex. They have now treated 75 patients and have not seen any severe veno-occlusive events after treatment of sickle cell disease. So also really remarkable. And the upper bar there, what you see, shows you the months after treatment. So with the longest follow-up here was 32 months. So again, also very remarkable results. Now, there's also been significant progress for other diseases. Here, just to list a few, I won't go into any details here. This is really just to list the different diseases. Here for immunodeficiencies, and again, there's just 3 publications here using a SYN-modified gamma retroviral vector or lentiviral vector, and with busulfan conditioning. And the figure here shows the T-cell reconstitution from the most recent study in the New England Journal in 2019. And you see it takes about almost 3 months though for the T-cells to really recover and reconstitute in the patient. There's also been significant progress for other diseases like the metachromatic leukodystrophy and adrenoleukodystrophy diseases, as you can see here, where they've also shown very efficient engraftment of these modified cells and improved outcomes. And there has been progress for patients with Fanconi anemia disease we've also worked on. And what's shown here is the study by Juan Buren's group from Spain. They saw a successful engraftment of gene-corrected cells without any conditioning. And although seen in the figure below, what is shown there is the overall results were a bit mixed. Some patients did show stabilization of counts and had some improvements. Others had less improvement with their counts. We did a similar study in Seattle with fewer patients and saw that pre-existing immunity may actually be limiting in some of these patients since no conditioning was used. So we did see immune responses against the VSV-G transduced hematopoietic stem cells. And this study, or the overall gene therapy studies for Fanconi anemia, are now being further developed by Rocket Pharma. So this is also an example where a collaboration between Juan Buren's group and our group ultimately led to the development of this gene therapy by a pharma company. Now, we had applied hematopoietic stem cell gene therapy in patients with glioblastoma, where we modified the hematopoietic stem cells to make them resistant to a combination of 06-benzalguanine and temozolomide, which allowed us to sensitize the glioblastoma to temozolomide and at the same time protect the blood cells. Also, these results were quite encouraging with the outcome improved outcome in these patients. We were able to administer more cycles of chemotherapy and improved outcome at 2 years with 43% versus 8% median, 8% survival in the historic control, which is shown again here on the small figure that's included in the slide here. We also saw the overall median survival was 20 months in our patient population. This really just a small study versus 13 to 14 months again in the historic control patients. So it was quite exciting that we were able to show some benefit in this setting as well. Now, while there's been tremendous promise for these gene therapies and gene editing approaches, there are significant limitations, and I will address some of those. Current approaches, as you know, require fairly large amounts of vector and gene editing reagents. And this could, of course, be improved by if we could target fewer cells, like maybe a more refined hematopoietic stem cell population, and I'll discuss that. Really like bone marrow transplants, all the ex vivo gene therapy studies require really highly sophisticated settings or institutions where that engineering and manufacturing can be done. So really this type of gene therapy would not be available to most people affected with hemoglobinopathies. Now, potential solutions here would be manufacturing in a portable device. And we've worked on that, and we'll discuss just very briefly in a later slide, like the Prodigy device, for example. Or, of course, if you could take all this into an in vivo gene therapy setting. But to address the first approach, again, all the current hematopoietic stem cell gene therapy studies use CD34 cells as target populations. And most of those CD34 cells are not hematopoietic stem cells that contribute to really hematopoietic reconstitution. So really, we waste a lot of reagents and, of course, also potentially increase the risk for insertional mutagenesis in cells we don't even need because they don't really contribute to reconstitution. So when Stefan Radke, or initially a German postdoc, now a senior staff scientist in my team, joined the lab quite a few years back now. The first thing I had asked him actually is to look into antibodies that are available to characterize human hematopoietic stem cell progenitor cells and really test whether those antibodies would cross-react with non-human primate cells. So then we could test these antibodies and hopefully identify hematopoietic stem cell populations or candidates in the non-human primate model that would allow us to really do longer, short, and long-term follow-up studies of stem cell modified cells. So he found that with only 3 conserved markers that cross-reacted perfectly based on in vitro studies, the CD34, CD90, and CD45RA markers, he was able to identify those three distinct populations shown here and labeled HSC, MPP, and LMP for multipotent progenitor cells and lymphomyeloid progenitor cells. And as you can see here as well on the right now, if we could just use the CD90 positive, CD45RA negative population, this population only made up about 5 to 10%, as you can see up here in the slide on the right. A bit hard to show with the mouse, but it's on the right side. That, of course, would allow us to have an enrichment of about 10 to 20%. And therefore be able to reduce the overall amount of vector needed for the modification of this more enriched cell population. Now, to see whether this cell population would really, how this cell population would behave in vivo, and how these cells would contribute to short and long-term repopulation. we marked them with different fluorochromes, with GFP, M. cherry, and M. cerulean, and then infused them into non-human primates that had received myeloablative conditioning, so we could really track exactly the contribution of these cells to short and long-term hematopoietic reconstitution. So this is shown here, and this was really one of really my most remarkable results and impressive results I've seen in non-human primates. You can see the engraftment. This is shown right now with the GFP positive cells in green. The engraftment came solely from this CD90 positive population. Again, this was only about 5 to 10% of the overall CD34 cells. Very early, as you can see here, around day 10, we saw a significant engraftment already. And then, most importantly, all the other lineages, again in green here, came from this CD90 positive population. So you can see the granulocytes, monocytes, T cells, B cells, NK cells, all the cells came from this population. And the MPPs and LMPs, really how we've defined them, they really barely contributed, or maybe there's a few blips here and there, but nothing really significant. So really everything came from this CD90 population, short and longer term. This is only 175 days, but I will show you also long-term follow-up data. Now, of course, we also switched to fluorochrome just to be sure it wasn't some odd thing with the fluorochromes. Here we used then the M. cherry to mark the CD90 population, and you can probably appreciate how similar that looks. Exactly the same finding with the different fluorochrome used for the CD90 population. So very consistent results, which again, it's hard to get that in non-human primates. And this is also again up to 175 days. But when we followed these animals, you can see here, this is more than 1,500 days now. So more than 4 years, you see the stability, especially with the red and green marked cells here. The amylin blue had the lowest cell number, so this is probably why it was a bit lower and a little bit less stable overall. But if we have now enough cells, now our cutoff is about 100,000 cells per kilogram of this CD90 population. That is really sort of the lower threshold. If we're above that, we usually get very good engraftment. Again, stable long-term, as you can see here. Now, Stefan and his team with Nyanada and Mark, they also did really extensive clonal tracking in these animals. Since we marked them with fluorochromes, we could really sort and analyze the individual cell populations really very thoroughly. So the different colors here indicate the contribution of the clones to the different lineages. So blue indicates clones that contributed to 5 lineages. So granulocytes, T cells, B cells, monocytes, and NK cells. And you can see that they start contributing very early, these 5 lineage clones, and then really within days. And of course, they stay around and take over now long-term in these animals, as I showed you before, 4 years. And it's also a presentation that Stefan gave at this year's ASGCT meeting in the presidential symposium. Okay. So then we, of course, also wanted to see whether we could use the CD90 population potentially for gene editing. This was done with a lentiviral vector, what I just shown you. And now we want to see, could we also do gene editing? And for gene editing, we were also interested in fetal hemoglobin reactivation. As I mentioned, of course, George Stamatianopoulos was in Seattle, and he's worked on this in this space for decades, as you can tell. And he was really the first one who described this concept, and here, as you can see, stipulated that the ability to stimulate fetal hemoglobin production in adults might be a therapeutic benefit in patients with sickle cell anemia at that time, or severe variants of homozygous beta thalassemia. And of course, you know, this launched really, I think, decades of research to cure these disorders with drugs, gene therapy, and now also gene editing. So there are really 2 main approaches for fetal hemoglobin reactivation. It's pretty simple. Either you inactivate the repressor, which is BCL11A, or you generate one of these hereditary persistence of fetal hemoglobin mutations, HPFH mutations, that will block the binding of the repressor. Either you inactivate the repressor or you block the binding of the repressor. And we focused on one particular mutation deletion, that 13 nucleotide deletion, which we knew could increase the fetal hemoglobin more than some of the other mutations in this space for the fetal hemoglobin reactivation modalities. And with this 13 nucleotide deletion, in vitro studies have shown that one can actually probably, in mouse studies have shown, one can probably get about 30% modification and increase. And 30%, 25 to 30% modified corrected cells, we know will likely be close to cure of sickle cell disease based also from allogeneic studies. So as I mentioned, we want to see could we use the CD90 positive cells again in addition to the CD34 cells. Here I'm just showing you how we did the CD90 positive, again, the gene editing approach for the fetal hemoglobin reactivation. So we got the CD90, isolated the CD90 positive population, modified only the CD90 positive cells, did co-infuse the CD90 negative cells as well, but only modified this very small fraction of CD90 positive cells. And then, as before, infused these cells into animals, non-human primates, that had received myeloablative conditioning, so we could really track very in detail the recovery after transplantation. So here we first, on the left-hand side right now, you see we were first tracking the percent of HBG promoter editing. So this is where we edited the binding site of BCL11A after transplantation. And what you can see here is you see both groups, also the CD34 edited cells and in blue, and then the CD90 edited cells in red. And you can see they're very similar. And again, and very stable for a long period of time. This is days now post-transplantation, in one animal now almost also 1,500 days, as you can see on that left slide here, left figure. Again, importantly, though, with the CD90 population, we had about more than tenfold fewer cells that we had to modify here, and we saw the same result. We then also, of course, measured the fetal hemoglobin in these animals, so HbF expressing red blood cells on the right side here. And as you can see, this is also very stable and very similar to what we've seen in the CD34 cells. So in both populations, the gray curves below that you see there was actually sort of the background fetal hemoglobin reactivation after transplantation, because every time you give radiation or chemotherapy, there's some fetal hemoglobin reactivation in animals, or for that matter in patients as well. And then, then as you can see, then it comes back down again. And so we were clearly significantly above this sort of early fetal hemoglobin background elevation. So now, this was done with CRISPR-Cas technology. Now, there are limitations, though, also now that we've learned and the field has learned with CRISPR-Cas approaches, because they do rely on double-stranded breaks, which has a number of potential side effects just listed here on this slide, including activation of p53. We can see large deletions or complete rearrangements and chromosomal translocations now, because we make these double-stranded breaks in the DNA. Now, fortunately, David Liu's group has developed the next generation technology that is called base editing, where we can now just edit a base instead of introducing a double-strand break. And so we can just modify a C to a T or a G to an A currently. And this allows, of course, for much more controlled editing and very low, if any, double-strand breaks using these base editors. Now, another really important aspect here is using these base editors and avoiding double-strand breaks is, of course, because we can now make multiple edits in one cell. I will show you an example how we will take or want to take advantage of this, but this is now also in the T cell editing space, very, very important, where people really want to edit multiple pathways, now checkpoint inhibitors. Again, we can now multiplex, do these multiple edits more safely in stem cells or T cells for different applications. So as I mentioned, it was important for us so we could also multiplex, and we remained interested in stem cell gene therapy approaches here. So one trend, of course, one important aspect currently is, as I mentioned already earlier, all the stem cell gene therapy studies currently are done with high-dose busulfan conditioning, which really most investigators are trying to also get away from the use of any alkylating agents or high-dose myeloablative conditioning. And you know, there's lots of attempts now trying to also use antibody-based conditioning, either antibody drug conjugates or just even the naked antibody to create some space without the use of any genotoxic alkylating agents. And so of course then it would be nice to have also in that setting the ability to, if the level modification levels are just not high enough, to be able to titrate that up and select for these modified cells in vivo. So that's one application why we're also interested in this multiplexing. But of course, the other interest is if we do this in vivo, direct in vivo gene therapy, in vivo correction of this of these cells, then we will likely need some enhancement enrichment step in vivo. And so because of that, we've been looking or been interested really in different ways to enrich after the fact, after transplantation or after in vivo gene therapy for these modified cells. And again, this just shows you the basic concept here. Now, then we could use a drug selection and increase the modification level of these modified cells. So for stem cells, we've been very interested in CD33 since we and others have shown that CD33 is actually dispensable for hematopoietic stem cell engraftment. And it can be targeted very nicely with CAR T cells or anti-CD33 reagents like Mylotarg. So there's been lots of research already done and lots of antibodies are out there now for potential use and have been used already in the clinic to some extent. So we've been studying exactly this approach. We want to now multiplex, induce fetal hemoglobin with one base editor, and then also knock out CD33 that would allow us then to do in vivo selection with an antibody against CD33. So we confirmed initially in vitro studies that we were able to efficiently modify both sites in bulk CD34 cells and then in CFUCs. You can see here on the right that most cells also did get both edits in blue, so CD33 edits and the HBG, that for the hemoglobin reactivation edit, again in blue. Of course, then we want to see whether these double edited hematopoietic stem progenitor cells can engraft. So we studied this in the immunodeficient mouse model. And as you can see here, in these studies, we saw comparable engraftment of these CD30 in the CD45 cells in between the non-edited and the HBG CD33 edited cell populations, again in the left lower figure there. We also saw a very efficient knockdown of CD33 in the below here in the CD14 monocyte population. So this is shown on the right lower panel there. CD33 expression again on CD14 monocytes. You can see a very efficient knockdown of this CD33. So the next question now was whether these cells with CD33 knockdown would be protected and could be selected for by anti-CD33 treatment with Mylotarg or gemtuzumab ozogamicin antibody. And this is shown here, where we saw very nice protection of these edited cells in green versus, again, in black, the non-edited cells. So we think this could actually work with this CD33 knockout for selection, and this would allow us to use a non-chemotherapy-based selection/enrichment strategy. Again, there's other people are working on this. Is not the only antibody-based strategy currently. People are working on approaches to use CD117 and CD45 for that matter. So there's a number of different approaches currently being explored by different investigators and, in fact, companies. Now, I want to switch gears just briefly and also talk about HIV, because we do have a large program in Seattle for HIV gene therapy and HIV cure. There are now almost 38 million people living with HIV around the world, and about 35 million have died from AIDS-related illnesses since the start of the epidemic. Now, with antiretroviral drugs, we have been able to control the virus, and that's really improved the life of millions of people dramatically. But we know these drugs cannot eradicate the virus. So we've not been able to cure HIV with antiretroviral therapy. Now, of course, this all really changed the notion of can we cure HIV with Timothy Ray Brown. And when he received an allogeneic bone marrow transplant, of course, the Berlin patient, for acute myeloid leukemia in 2007. So he received a transplant from a donor who was also resistant to HIV because of a CCR5 delta 32 deletion that naturally occurs in some patients, in some people. And he stopped his HIV drugs right after this transplant and never had a rebound. So it was the first person who was actually lived HIV-free without any antiviral drugs and was cured of HIV. Now, of course, the question was, do you really need these CCR5 knockout cells or not? And that was in the Boston study. So they repeated pretty much the same study, just not using these CCR5 delta 32 cells. And that didn't work. So it was quite clear there's something now to the CCR5 delta 32, so the HIV resistant cells. That was obviously important. And so there's now the Berlin patient, of course, there's the London patient, and then there's another patient that was just recently reported as well, and probably a few more that we just haven't heard yet. So this seems to be working, but of course, this is a transplant, an allogeneic transplant for malignancy, which would certainly not be anything that we could offer to people living with HIV. Now, because again, we know we needed these CCR5 negative cells, and of course, we've been interested in engineering, gene therapy engineering, and of course, in the stem cells. We have had several large projects in Seattle, and that's why I just want to present this briefly, and that brought together a number of different groups to develop approaches in the autologous setting. So we could modify the patients. You know, we've done all this in non-human primates, but ultimately the idea is really to do all this in patients, taking the cells, modifying them such that we can knock out CCR5, but then also ideally really knock in or modify them such that they would also provide these sort of anti-HIV properties that we saw in the allogeneic transplant setting, and that were likely also very critical for the cure. So again, this is just one of these larger projects we work with, again, with Magenta here, to also now have a non-alkylating technology, antibody-based technology, to condition the patients and for the infusion of genetically modified cells that would then protect the patient. In our case, again, these are non-human primate studies, the monkeys, from HIV or SIV infection. So now we can modify the hematopoietic stem cells here, and we also know that if you modify, of course, the hematopoietic stem cells, we can also modify stem cell derived T cells. And then we can also certainly now incorporate a CAR construct, so we could generate stem cell derived chimeric antigen receptor carrying modified T cells or virus specific TCRs. We also know we can modify B cells with stem cell derived B cells. We could, of course, then also express broadly neutralizing antibodies for a long period of times, or even antibody-like molecules like ECD4-IG for longer periods of time. So these are just studies and approaches we've been pursuing in Seattle with collaborators. So here's just one of these collaborations where we showed that we can indeed generate stem cell derived CAR T cells against HIV. This was a collaboration with Scott Kitchen and Jerry Zak from UCLA, and this resulted in a number of publications just to show that we can indeed generate these stem cell derived CAR T cells and CAR NK cells, in fact, and CAR monocytes. And that these cells, when we did a longer follow-up here, that these cells really provided really multi-lineage engraftment. So, and this was a long-term follow-up with 2 years. This is the follow-up study. And that these cells really now, the stem cell derived cells, CAR T cells, would traffic to the reservoirs, just shown here schematically what we showed in that paper, to the lymphoid germinal centers, the gut, and the CNS, which is of course very important for HIV because there's concerns, and we know HIV can also be in the CNS. So there's always been a very large interest in being able to also eradicate HIV in the CNS. So, however, as I mentioned before already, access to these really promising genetic medicines is really highly limited. And probably of these, the 18 million people living with sickle cell disease globally, people, not just us, companies, Bluebird, Vertex, estimate probably only 25,000 patients, or maybe 50,000, less than 1% will have access to current gene therapies. So would not be a lot of patients, some patients, even again with Zynteglo. So because of that, next generation technologies need to be developed, and we've been thinking a lot about that. And on the left, what you see here is now what I mentioned briefly before is, yes, we could do everything in a prodigy setup, and we've talked last night quite a bit about this with Christopher and with some of the people I met yesterday. And that, I think, is a good next step. We could do everything in that and maybe combine with, again, conditioning or the next generation conditioning. But what if we could just do this also in vivo? Could we get to a point where we modify the cells directly in the patient rather than taking the cells out? And of course, if we could do this directly in patients, that might facilitate and really make this much more accessible than again, any ex vivo approach. Then we could really manufacture the nanoparticle or vector in one setting, in one place, and then really ship it around the world, and then hopefully with an injection, maybe 2 injections, correct the disease or treat the patient. Now, of course, AAV has been used for in vivo gene therapy, to treat spinal muscular atrophy and inherited retinal disease, or to deliver gene therapy machineries in vivo to the liver, but to my knowledge, not to stem cells yet. But this is also a collaboration we've started with Hildegard, and Chris and my team, they just received actually a fairly good sized grant to really study now B cell editing in vivo and also stem cell editing. Now, we've shown in the past, quite a number of years ago now, that we can also use cocos-pseudotype lentiviral vector or foamy viral vectors to correct severe combined immunodeficiency. And this is done in a dog model that's very similar to the human severe combined immunodeficiency. So all we did is we had a foamy or lentiviral vector expressing the common gamma chain and injected that vector into very young newborn dogs affected with immunodeficiency, with SCID, so within the first 4 weeks. And as you can see on the right, it was actually quite impressive. We see very, very early T-cell reconstitution there, much earlier than when you do this ex vivo. So certainly in that setting, that could also be an approach move to the clinic by simply providing in vivo gene therapy. Of course, all the cells lack the common gamma chain. In this setting, again, you've got this dramatic selective advantage of the corrected cells. So you don't need any additional selection or enrichment step in vivo, because really only the corrected cells really take off in this setting. Now, we have worked in Kurt in my lab, a graduate student with Stefan, also on pseudotyping lentiviral vectors, now with measles envelopes. So either measles or a Nipah from the measles family. But what we've been wanting to do with this approach now, again, using measles envelope allows us to incorporate a more targeted approach. So we've been very interested, that gets back to the CD90, targeting these more refined populations. We've been very interested in targeting the CD90 population in vivo. We've now been able to generate actually a single chain variable fragment to directly target also CD90 cells in vivo with a viral, the pseudotype lentiviral vector, again, using a measles envelope. So that is also progressing in our lab as one approach to target in vivo now this CD90 population. Now, in a really fantastic collaboration with the David Baker Lab in Seattle, we've also been working on the so-called co-locker system. Using this co-locker system, we can be much more specific for cells. And of course, he's already shown this for T cells and the CAR T cells on the left-hand side. This is actually what he's already published with Stan Riddell here in Seattle as well. So you need 2 unique antigens for this CAR T cell to bind. This is the basic concept of this co-locker system. And of course, we want to use this system with David so that we can now target not only one CD90, which is also on mesenchymal cells, for example. So we want to really have target cells much more specifically, for example, that there's now a CD34 and a CD90 antigen expressed. So just to show you, there's different ways to become more and more specific in our targeting approaches in vivo. Now, this was viral based. There's a lot of effort in Seattle and our groups and really around the world, I think, using different types of nanoparticles. We've been interested in polymer-based nanoparticles, mostly because Matthias Stefan there has developed a platform for T-cell therapies using polymer-based nanoparticles. Then, of course, Jenna Dare, who was also in my group and has been having her own lab for a number of years, has developed these gold nanoparticles, which is also a very attractive novel approach to can deliver gene editing technologies or tools to stem cells and potentially other cell populations. And then, of course, we've all heard about the lipid nanoparticles. Drew Weissman has really been a pioneer in that space, and we've got also a number of really very nice collaborations with Drew, who's just a wonderful collaborator, looking at the potential of lipid nanoparticles for both targeting T cells and stem cells in vivo. Another really great collaboration I do want to mention here has been with Andre Lieber's group in Seattle. We both really had worked with George Stamatianopoulos for many, many years. And of course, now we've had large grants now focusing on gene therapy for hemoglobinopathies. And as many of you know, Andre has really been pioneering this helper-dependent adenoviral vector system, a gutless vector system. And then about 5-6 years ago, we started collaborating to see how could we move this at least beyond the mouse system, also in the non-human primate system. And of course, listed here are the unique advantages of this system. You can modify the knob, so you can actually specifically target these CD46 expressing cells, and it happens so that CD90 positive cells have the highest expression of CD46. So we fairly, preferentially target these CD90 positive cells with this fiber. And then, of course, there's episomal delivery with CRISPR-Cas base editors or prime editors, and we can integrate also payloads using the Sleeping Beauty system that can also be delivered with this helper-dependent adenoviral vector system. So really, overall, a very beautiful system to kind of do it all. And here, just a few data when we started out doing this using his vector system. So we included this MGMT P140K selection system that we also used in the glioblastoma study, and had really extensively studied ex vivo in our non-human primate models and dog models for the enrichment step. This requires a low dose of BCNU, a fairly low dose of BCNU for in vivo enrichment and selection, and that transposase Sleeping Beauty system to integrate that cassette, of course, into the genome. So then with this approach, with this adenoviral vector, this is just here using a GFP vector with this MGMT cassette. You can see initially without selection, the foothold, the initial foothold is lowish. This looks fairly low here. It's typically probably anywhere between 2, 3, up to 5% if in non-disease models. But then we can very nicely select up, as you can see, with a few doses of that dose of selection to really high levels, certainly therapeutic levels, as shown on the right there. Now, in a thalassemia intermediate mouse model with an erythroid-specific promoter, you can see actually higher initial engraftment here, and then because of the survival advantage of the corrected cells, we actually see then again already initially higher foothold. Then we can also select to the desired levels with the selection enrichment steps in vivo. As you can see then again with the red arrows there, and the levels go then way up to more than 60% modification levels, and leads to the cure of this thalassemia in intermediate mouse model. We've also shown this using now CRISPR-Cas delivering the again editing to cure the sickle cell disease in a sickle cell disease model. As you can see here, again, we can deliver to transposase system. We can also multiplex and also use CRISPR-Cas using an integrating delivery of a gamma globin gene plus reactivating fetal hemoglobin. So this is multiple things can be done with this adenoviral vector system. And just to show you here a multiplex where we delivered actually a gamma globin gene plus fetal hemoglobin reactivation, which led to the essentially cure of sickle cell disease. When you look at the spleens there on the left, very left, of course, is a normal spleen, and the right are the treated spleens, and the big spleen is the diseased spleen in the mouse model. So very efficient approach. We can do the same thing with base editing, so we don't even need the CRISPR system. So again, very efficiently able to deliver CRISPR-Cas technology, base editing technology, plus any sort of enrichment steps. And as I mentioned before here, we use this low dose sort of the 0.6-benzalkonium BCNU approach, but we're also working on combining this with non-alkylating agents now. And really what we've presented, Andre's lab really also at the last ASGCT meeting, we can deliver really very complicated and complex prime editor constructs. This was again another collaboration with the David Liu lab, and clearly, because not all, as most of you know, not all mutations can currently be tackled with base editing because the technology only allows right now change from a C to a T or A to a G, but not a T to an A to correct the sickle cell mutation. So with prime editing, you can do that, and that's exactly what we've actually shown with prime editing with David Liu's group. We generated, put together a really very complex, highly optimized 16 kb prime editor to directly fix the sickle cell mutation, and so an almost correction in the sickle cell disease Towns mouse model again. When you look at the graphs on the left lower corner there, you can see in the left you see the Towns mouse there, the spleen sizes, and then significant reduction in the spleen size after prime editing in these mice. And then the same thing when you look at the percent sickle cells on the left lower corner there. The orange versus the green. Again, orange is the disease model versus then the correction in the green. Okay, so this platform we think is quite powerful, and because it's so flexible, we can insert a gamma globin gene with an optimized large regulatory element, again up to 35 kb. Can use CRISPR-Cas technology, base editing, prime editing. So we certainly think we're quite flexible with this tool. We've of course looked in non-human primates. As I mentioned, that's when we started about 5 years ago, collaborating more closely together. And because of this fiber, it's a different, the AD35 fiber we detarget from the liver, which of course has traditionally been a concern with adenoviral vectors. So what you see on the left there is with the AD35 fiber, we do not transduce the liver. So this is shown with that GFP vector on the left, but really also on the right, just looking at the liver functions, we do not see any increase in liver function abnormalities. This is in the mouse with that AD35 fiber, because again, this is sort of a detargeting from the liver. We've also looked in the monkey. We've looked for efficiency, and was actually very efficient when we mobilize the cells into the peripheral blood with regular mobilization agents, GCSF and AMD 3100 or plerixafor. We hit about 50% of the circulating stem progenitor cells. So the cells, if you take the CD34s out after mobilization and transduction and then plate them about at 2 and 6 hours, about 50% of these cells are green, from green colonies. So very efficient transduction of the cells. Now, of course, not all these cells home back to the marrow, but at least we could show that the initial transduction is very efficient. Again, only very mild transient liver abnormalities when we use this approach with the modified fiber. And we've shown also very efficient again, gene modification in the monkey. Just one monkey shown here. Again, after selection, we can titrate up the modification levels to very high levels. And we've shown that this is safe in the monkey. And safety studies we've done now, the company as well in SOMAG, and they've taken this on to further develop this in more than 14 monkeys, that this is really safe when we infuse this vector, the AD35 modified vector. Okay, now just
we only got 5 minutes left, unfortunately.
Yep, almost done. Almost done. This is wrapping it up here. Thank you. Now, as I mentioned, we're also now coming back. We've been working very closely with the Gates Foundation to make this also develop a platform for HIV. What we've done with ex vivo, we think we can do all of this in vivo. Now also we can knock out CCR5 and then endow these cells in vivo, again with either broadly neutralizing antibodies or ECD4IG, or even provide this CAR construct in vivo to modified cells directly in vivo with again anti-HIV properties, very similar to what, of course, cured the Berlin patient Timothy Ray Brown. One of my last slides here was very encouraging for us to see that how Francis Collins highlighted really in vivo gene therapy at our last ASGCT meeting. He's done this now a number of times and talked several times that how he thinks that this is really sort of referred to it as a major step forward. And I think this is the conclusion here. We think the CD90 population is really, feel like it's a very good target for ex vivo and in vivo targeting. Base editors can be used to multiplex. I think we all know this for stem cells and T cells. We do think there's now a number of really encouraging platforms for in vivo delivery as well, to modify cells directly in vivo. And then of course the adenovirus platform, I do think is also quite flexible, and it will also be further developed by us in Seattle, but also by again the company. I think I'll skip over these. I just want to show again how gene therapy has also just really progressed over the past, 5 to 10 years. Just the number of therapies currently in the clinic, as you can see, for rare diseases and anti-cancer, 362, 360. And this is really very encouraging that there's a number of INDs now. How look at the curve, how this is, and it continues to go up, the interest in the space. The number of approved gene therapies now with Carvykti most recently, is Integlo most recently. And then of course the landscape. And I think this is my last slide, Christopher, just the enormous interest in cell and gene therapy, as you can see with all the different companies. With this, I'd like to, of course, thank my own team, my program, many of the fantastic collaborators, and of course, the Vivo Lab, the Adair Lab, the Walter Lab in Seattle, and many of the other collaborators over the years. I have to really thank David Liu, who's been just a wonderful collaborator, and also Drew Weissman, and that's it. With that, I think I'll finish and conclude. Thank you very much for listening.
Yeah, thank you very much, Hans-Peter. Big applause from all of us. These are really brilliant data, very exciting to see, especially for those who have been in the field for many, many years now, to see this progress in this holy grail of in vivo targeting and in vivo selection. Both seemed impossible for many, many years, and now you seem to be getting there. And I would also like to remark that it's highly important to have that broad arsenal of modalities available, from several viral vectors up to nanoparticles, because sooner or later there will be components in the delivery system, also in the nanoformulations, that may elicit immune responses. So it's very important to have different choices available also for patients that may require more than one treatment in their life. Unfortunately, we've run a bit out of time, but it was really important that you took all the time to present what you have developed over the many, many years with your collaborators and in your fantastic lab. So I think we simply have to give up on questions at this point, because we have to close sharply at 1 o'clock. But you'll be around for a chat opportunity for those who would like to take more time to talk with you today. And again, I would like to mention that we are really looking forward to hope to see you more frequently in Berlin in the next couple of years, when we can have you here as a visiting professor of the BIH. So again, many thanks from me on behalf of the entire audience for this great, fantastic talk. And I think this really has been highly inspiring and will have a big impact also here in the Berlin community. Thank you, Hans-Peter.